Thank you all for coming here this morning. The woman before you, the one on the mask, her name is Carly Koch, and she knows she's dying. She has a severe immune deficiency, and the severity of the disease is of a nature where, as you can see in the picture on the right, she and her mother have started to make end-of-life plans, like who gets her things, how she wishes to be buried, what music to play at the ceremony, etc. It's hard to begin to think about what that must feel like, especially when you know what's happening. And what's happening is she has a loss of function mutation in DOC8, and we know where DOC8 lives. There it is on the short arm of chromosome 9. So my central message to you this morning is, over the next decade, the clinical landscape of dealing with folks like Carly will fundamentally change. You know, we've witnessed over the past couple of centuries what's happened to infectious disease. If it's caused by a bacterium, then for many of them there are antibiotics that can be curative. If it's caused by a virus, then many of them can't be directly cured, but they can be robustly prevented with vaccination, and polio is a prime example. And I'm not going to stand here this morning and say to you, in 10 years, every genetic disease known to humankind shall be eradicated by genome editing. But there is no question, none whatsoever, that over the next decade, we will think clinically about how to manage genetic disease and other diseases in a fundamentally new light. And genome editing will play a major role in it. And so what I'll do over the next couple of hours is tell you what we're doing clinically and preclinically at Sangamo, and give you a sense of what's to come. I respect the fact that many of you are not practicing clinicians, and genome editing therapy for immune deficiency due to DOC8 deficiency is not on your tomorrow's agenda. What I'm hopeful you can take away from this is an appreciation of the extent to which work all of you do or plan on doing has and will contribute to the faster adoption of these genome-directed approaches translationally. Because, as I'm going to point out again and again throughout this morning, so much of what we've been able to do clinically and preclinically over the past decade derives and is based on work that in and of itself was not directly focused on putting things into the clinic. If there's one thing I'd like you to take away from this morning, it's on the next 2 slides. It is that genome editing has changed the definition of the term druggable target. One thinks of druggable targets as kinases, such as BCR-ABL, which is targetable by Gleevec, or, Advil, ibuprofen, prostaglandin metabolism, small molecules that inhibit active sites of enzymes. The new definition post-genome editing is that if it's in the DNA, it's a druggable target. And lest you think this is hyperbole, I will show you this morning how we've reduced this notion to practice in the clinic and in discussions with the Food and Drug Administration that have approved investigational new drug applications for approaches that really are not tractable, to the best of our knowledge, using any other path other than genome editing. To give you a short sort of summary landscape of where we are with respect to DNA as a target in the clinic. I'm going to focus today exclusively on somatic approaches and spend no time whatsoever on the much recently discussed germline therapies. First, because I had the good fortune of expressing my strong opinion on the matter in an article that came out recently. And second, because we have a very distinguished second speaker today who will cover this issue comprehensively. I'll focus exclusively on DNA-based approaches in the soma. And to a very first approximation, the way to think about this is you can think of traditional gene therapy, which is typically a virus, which delivers a payload to a cell or a tissue. and the payload integrates randomly somewhere in the genome. And some really exciting progress has been made in this regard over the past 20 years or so since this was first tried clinically. But I won't spend any time talking about conventional gene therapy today and focus exclusively on genome editing for obvious reasons. One can sort of think about two separate ways of deploying it. One can take a subject with a condition, do genome editing on her or his cells ex vivo, and then put the cells back in. And I'll tell you about our work preclinically and clinically that we've done for HIV, and also about our preclinical and advanced non-clinical work for the hemoglobinopathies, so beta thalassemia and sickle cell disease. I'll also talk about approaches for genome editing in vivo, in which no cells are taken out. Instead, the genome editing machinery is delivered to the individual. And I'll describe to you our work on hemophilia and the lysosomal storage diseases. What I'm hopeful you will take out from all of this is an appreciation of what it takes to take a double-strand break and then deploy it in the real world of a clinical trial. I'm about to show you one of my favorite pieces of data of all time, which unfortunately is not from my group. What would I have done to have done this experiment? It's really beautiful. And it's striking because it reminds us all that the way that genome editing has taken over the world, 15 years ago, it was not exactly on everyone's agenda. Today, as I often joke, first-year graduate students, when they join the lab, get given a Gilson and an expression plasmid for Cas9. The reason this happened is because, of course, double-strand break repair is so ancient and so powerful. And this is a yeast result. This is from the Resnick lab. And what they've done here is taken Cerevisiae, our little biologist's best friend, and run the chromosomes out on a pulsed field gel. So these are chromosomes. So this piece of DNA is 1 million base pairs, this piece of DNA is 660 kb, and so on. And so then they irradiate the poor things, which of course, as you know, radiation induces double-strand breaks. And what happens, of course, not surprisingly, is after the more you zap the poor things, the more the genome is shredded. So you go from this beautiful chromosomal complement to this devastation zone. So then you turn off the radiation source and you just leave the yeast there to sit there and just sit there. And over the next 2 hours, this little thing puts the genome back together again, using the biological process that genome editing relies on, both in your basic research and in the clinic, which of course is homology-directed repair. Moving across hundreds of millions of years of biological evolution, all the way to you and I, you're all familiar with the fact that double-strand breaks create a chromatin signature. And this is a study in human cells in which cells were just incubated or irradiated with increasing doses of double-strand break-inducing agent. And there's a break, and there it is 2 hours later in a human cell, and 4 hours later it is gone. And here's a cell with 2 breaks, and there the breaks are still, and 4 hours later, they are gone. I don't have a way of telling you if this was HDR or NHEJ, but from yeast to human, cells love repairing breaks. It's what they do. And that, of course, is what we collaborate with when we engineer a nuclease, whether a zinc finger, a tau effector, Cas9 guided by an RNA, a meganuclease, a megatau. Your break-inducing agent invokes a natural process that's ancient and powerful. And so for us, over the past 15 years of working on this, we've had this marvelous journey through collaborating, if you will, with Mother Nature and the cell to try to understand how to best invoke DSBR to attain the translationally relevant editing outcome that is relevant to the specific condition that we wish to treat. Before I dive into clinical applications of editing, probably my second most important slide this morning is to re-emphasize a point that is familiar to you, which is the extent to which the development of editing is a triumph of basic research that I think, having worked 15 years in industry on this, deserves to be compared to the discovery of restriction enzymes and recombinant DNA technology. All of you know that restriction was not discovered because anybody wanted to make a plasmid. It was discovered by Salluria. The phenomenon was that certain phage don't grow on certain bacterial strains, and fast forward to the late 60s, early 70s, when, of course, Dan Nathans, Werner Arber, and Ham Smith, and Stan Cohen, and Herb Boyer, and all of those people took this naturally occurring biological machine, restriction enzymes and ligases and plasmids, and there we go. We now have, recombinant insulin, recombinant EPO, Herceptin, and what have you. So Salluria was not working on recombinant DNA technology when he discovered restriction. So the folks on this slide are sort of the four titans of the academic effort that contributed to editing, and I think it's critical that they be recognized as such. Maria Jason and Dana Carroll, of course, who's here in the room, have done some path-breaking work illustrating what a double-strand break when targeted can do. And Aaron Klug and Carl Pabo, with their work on the structure and subsequently design of zinc finger proteins, enabled us and a wealth of scientists in the academic community to then target these breaks to endogenous loci at a specificity and efficiency that made the development of genome editing possible. And of course, essential on this slide would be a picture of Ulla Bonas, who discovered the TAL effector code, which has been, of course, so wonderfully useful in the design of talons. And last, but putting it very, very mildly, not least, Jennifer Doudna, whose discovery of the fact that Cas9 is an RNA-guided endonuclease, of course, brought you all here, because it took editing and everything we've known about it and converted the rules of approaching your locus of interest to those discovered by Watson and Crick. At Sangamo, 13 plus years ago, inspired by all the work that was done by the folks you see on the screen and others, we set out to deploy genome editing in translational settings. And this is a key opportunity for me to acknowledge in the strongest possible terms my wonderful current and past colleagues at Sangamo, in particular Michael Holmes, Philip Gregory, Jeff Miller, and Ed Rebar. Working shoulder to shoulder with them for the past 15 years of my life has been an absolute highlight of my entire life, and I would not be here today if it wasn't for them. So over about 6 or 7 years of focusing on genome editing of endogenous loci, primarily in human cells and primarily for therapeutic applications, we discovered that the designed nuclease of our choosing back then, 10 years ago, and to this day today, the zinc finger nuclease, can be used to drive targeted correction in 2005, integration in 2007, disruption in 2008, and more elaborate processes such as larger deletions, integrations of multiple transgenic cassettes, etc., such that by 2010, we essentially knew that the age in which the genome, the human genome, is no longer experimentally manipulable, that era was over. It was done. The genome stood no chance against the power of the break and the genome editing toolbox. The outstanding question in the field was the facility of deployment and the speed of adoption. And two important developments then transpired, like as I said, Ulla Bonas' discovery of the TAL effector and the code, and of course Jeff Miller's and Ed Rebar's work at Sangamo building the first TAL effector nucleases that worked on endogenous loci. And as all of you know, TAL effectors are more straightforward with respect to design and assembly than zinc fingers. And then what was exciting to us and everybody who worked on the field is every single thing we learned over 8+ years of work on editing with zinc fingers was immediately, like that, portable to using TAL effector nucleases. Why? Because they rely on the same biological process, double-strand break repair. You just need to invoke the break efficiently and work with the cell in all other aspects. Everything we developed with zinc fingers assays, approaches, constructs was portable to TALS. And then, of course, Jennifer's discovery of Cas9 made things even more appealing for the entry user, if you will, because one could address a break to an endogenous locus via, as I mentioned, Chargaff rules, so Watson-Crick base pairing. But again, as all of you know, everything we learned about the toolbox of editing that you see on the screen that appeared between 2002 and 2010 was robustly copy-pastable to the toolbox of editing with CRISPR, with Cas9 guided by an RNA. So I will speak today exclusively about zinc finger nuclease-driven editing because this is the platform of choice for clinical applications. But as I've said over and over again this morning so far, you should be inspired and encouraged by the fact that you are break-driven editors. And thus, whatever you hear today about zinc fingers, you can do the internal global search for place with respect to your nuclease of choosing, which I suspect at this point is Cas9. In preparing this talk, I needed to try to mentally jump back to 2001, 2002, when the only two genetically tractable eukarya were yeast and mouse embryonic stem cells, and hence mice via conventional gene targeting. And the rest of organismal and cell biology, the genome was not amenable to being addressed in a targeted fashion. At the time, conventional gene therapy, which is taking a cell, integrating a virus randomly, and then putting the cell back in, was making important strides. And one of the early successes, which was an enormous event, came from Alain Fischer at the Hôpital Necker in Paris. What they did is they took several children with X-linked severe combined immune deficiency, also known as bubble boy disease. And the original bubble boy is David Vetter. He lived in a bubble because he had no functioning immune system, and he only went outside a few times in a spacesuit that was specifically designed for him by NASA. He ultimately died during a bone marrow transplant that failed for reasons unrelated to gene therapy. But by the late 90s, early aughts, it became fully feasible to do conventional gene therapy. So take a child with this condition, harvest bone marrow, which contains hematopoietic stem cells, infect those cells with a virus that randomly integrates the corrective transgene, and then put the cells back in. And in 2001, 2002, the field of gene therapy and the field of molecular medicine was thrilled at the report of robust efficacy. The children went from a complete failure to thrive, massively disseminated infections. Boys with X-linked severe combined immune deficiency lead a very, very difficult life. And it's essentially, without a transplant and without living in a bubble, the disease is lethal. So there was these wonderful reports of efficacy which were thrilling to everyone. I vividly remember that. And of course, as many of you know, this was rapidly followed by the report of a iatrogenic event, so a severe adverse event on the clinical trial, in which several of the children, forced to be specific, developed leukemia. One of the children died. And we know why that happened. That happened because the payload delivered by the virus I mentioned to you integrated randomly, and in those individuals, it integrated into and activated a proto-oncogene, hence the leukemogenic event. And so at the time, this is a sentence from a paper that appeared at the time, a thorough reassessment of the potential risks of retrovirally mediated gene therapy is warranted. And we realized as we were developing genome editing of human loci that that's where a legitimate unmet medical need exists. Here's a severe disease that needs to be treated that's caused by a mutation. Conventional gene therapy is being reassessed because the safety profile appears to be at that point clinically unacceptable, although much good work has been done since to improve the situation. And so we thought, well, enter genome editing, and here's a simple flowchart. We're just going to do genome editing for this. We have the disease, bubble boy disease, and we have the gene target, IL-2R gamma. We'll just build a nuclease. In our case, we'll build a zinc finger nuclease, because that was the one we knew how to build, and to this day we use because of clinical potency and specificity. And then we'll just induce a double-strand break at the target, and we need to develop delivery that would be sufficiently robust. It would have to be safe. And of course, we have to do this at good manufacturing practice standards, which involves a very substantial amount of work. And then in this case, we would probably need to invoke a correction of the mutant allele. And there we go, we'd have a disease therapeutic. What could be simpler? Well, so this morning, you'll hear about what it took to make some of these kinds of things happen, and sort of what we knew and what we didn't know, and what we've learned and what we're taking forward. I'll walk you through the specific case of bubble boy disease as a case study, because there's a number of, if you will, teachable moments that we've taken forward subsequently. So the first one to begin with is that bubble boy disease is monogenic, so it's caused by mutations in the same gene, L2R gamma. But the problem is that there are multiple different mutations that cause it. And at the time, how efficient targeted integration would have been was not entirely clear. So we chose to go for correction. But the problem was, of course, correction of what? There are many different mutations that cause this. We noticed in Jennifer Puck's very useful schematic a mutation hotspot in exon 5, and we went, well, maybe that should be the one. Well, next step is induce the break. Well, where do we induce the break? So I mentioned Maria Jason in one of my opening slides. As I'm hopeful all of you know, and if not, please allow me to recommend this as an essential reading for any genome editor. One of the foundational findings in this field is that gene conversion tracts from SDSA-driven HDR are quite short. And translating that from the acronym to the English, cut as close as you can to the position you wish to correct, as close as humanly possible. And this is a beautiful result from Beth Elliott and Maria Jason that shows that using a reporter gene, I think in mouse cells, and we've done very extensive studies like this in human cells, both primary and transformed, and the conclusion stays. Gene conversion tracts from a break in mammalian cells are relatively short. I also find it fascinating that, as Dana showed very conclusively, gene conversion tracts from breaks in Drosophila are longer. Which, of course, raises all sorts of wonderful questions as to what explains the interspecies difference. Can we exploit it? It's good stuff. But back then, which was 2003, we knew we had to cut at the mutation. So I'll spend 3 or 4 slides telling you about zinc fingers, fully understanding that all of you have Cas9 in your shirt pocket or in your purse, and that you probably are not going back to lab tonight to use a zinc finger. The reason I want to do this is to highlight for you what it took to take a designed nuclease to the clinic or into more challenging primary cell or living organ applications. Because as all of you over the next 30, 40, 50 years of your professional life advance editing to your favorite application, I think it might be useful for you to think about what it took as a case study with a zinc finger, and thus think about porting those kinds of efforts to your own favorite nuclease of choice. The zinc finger, of course, is the most common DNA interaction motif in eukarya. This is a beautiful crystal structure that was solved by Carl Pabo in 1991. Here is a 3-finger protein recognizing its target 9 base pair sequence. And over the next 2 slides, I'll zoom in onto 2 separate alpha helices recognizing 2 separate triplets. We'll first zoom in on this one, recognizing that triplet, and then on that one, recognizing that triplet. And you will immediately find something quite striking, which is that the geometry with which the alpha helix docks in the major groove and recognizes 3 base pairs of DNA is extraordinarily similar, despite the fact that these are 2 different amino acid sequences recognizing 2 different triplets of DNA. And so to illustrate that point, here is this alpha helix, RSDELTR, recognizing GCG. And here is a different alpha helix, RSDHLTT, recognizing GGT. This similarity with which the alpha helix docks with the triplet, and this quasi-linear arrangement in which specific amino acids recognize or appear to specify particular bases in the DNA, localized largely to one strand of DNA, inspired Carl to write this rather famous sentence, which has been tremendously impactful in the history of genome editing. I think it's fairly safe to say that if Carl had not proposed this, I certainly would not be standing here today. And what Carl said is, this structure provides a framework for understanding how zinc fingers recognize DNA. and suggests that this motif may provide a useful basis for the design of novel DNA binding proteins. This was in '91, and to say that this is an understatement would be an understatement. This proved to be oh so true and oh so useful. And quite strikingly, what I find really fascinating is I have the honor of knowing Carl quite well, and I've spoken to him a number of times about this. His initial quest to solve this crystal structure was not motivated by the desire to develop genome editing. So a wonderful example in which very basic biology, high-resolution protein-DNA interactions resonated in highly unexpected ways. I will go through the next few slides really quickly because, again, I respect the fact that you do not have zinc fingers tattooed on your hippocampus. But just to give you a sense of what it is that we can do with them that is translationally useful so that you can think forward to what you do in your daily practice. The elementary binding unit is a single zinc finger. It's short. It folds via coordination of zinc. It recognizes 3 bases of DNA. And specific alpha helical residues determine triplet binding preference, as you can see here. We have a platform, which we've spent the past 20 years building. It's actually quite large and quite robust. It uses 2 finger modules for design. I will spare you the technical rationale for why that is. Just trust me, this is the way to do it. And when we wish to address an individual locus, if you're working with Cas9, there are multiple algorithms to use Chargaff rules, basically to design a guide against your target region. When working with zinc fingers, you basically select a stretch of your target locus, then you pick individual modules that specify the hexamer sequences that the target consists of, and you string them together. And there you go, you have a multi-finger protein that specifies your target of interest. So far, all you've done is you've recognized a locus in vitro. Now what you have to do is recognize it in vivo, in heterodimer configuration, to induce a break with high efficiency and specificity and attain the editing outcome of your choosing. So this diagram, I think I borrowed from a review Dana wrote, and it really highlights the known unknowns that sort of lurked before us when 12 or 13 years ago we wanted to do correction of a mutation in IL-2R gamma. So it was clear to us that nuclease biochemistry is limiting. We have to be able to induce the break with maximal efficiency and specificity. And the solution to that is optimization. So the zinc finger is unique among designed nuclease platforms in allowing an extraordinary range of biochemical tuning. It's like a rheostat, as you're about to see the data, which is why it's been so powerful. We knew that resection tracts are short. We knew that from Maria's work on reporters. We subsequently proved that on endogenous loci in multiple human cell types. So we had to be able to place the cut as close to the mutant position as we possibly could, and we succeeded in doing that. But so far, all you've done is you've induced a break very efficiently close to the mutation. We all know that SDSA-driven HDR evolved to use the sister as the template. It did not evolve to use anything we've co-delivered, an oligo, a short PCR product, a plasmid, a virus. It evolved to use the sister, which is next to the broken one. So we have to be able to compete with it. And furthermore, we also know from a wealth of work that's transpired, starting with the initial discovery of SDSA-driven HDR, that cell cycle state matters. So we've spent, it's kind of surreal, we've spent the past 13 years thinking about delivery and cell husbandry. Attaining the biologically relevant outcome of your choosing with editing, whatever your nuclease platform, requires a detailed understanding of how to get the payload into the cell and how to make sure that the cell resolves the break via the pathway of your choosing. And an enormous amount of work has gone into this. And I will report some of this to you today, and I'm hopeful some of it will be useful to you. Last but not least, you realize this was 2003. Today, undergraduate students can order a lane of deep sequencing on a HiSeq by one, snap of their finger, and nobody notices. In 2003, there was no deep sequencing. There was Sanger sequencing, which is, not exactly high throughput. So we needed to build an assay for gene correction that was highly linear and had an extremely low false positive rate. And so that we proceeded to do that. But this, of course, has since been superseded because, as mentioned, deep sequencing is the way to do this. If you have access to a deep sequencer, that's what you should do. Let me restate again something that you've heard from me this morning and at dinner with Dirk Hockemeyer last night. I know he mentioned this to you as well. If your goal is correction, place the break as close to the mutation as you humanly can, literally. If you can, now there are some interesting points of nuance here which we can discuss. In particular, one major point of nuance is once you've performed the act of gene correction, is the chromosome that you have made recleavable by the very same nuclease that you've put in the cell to invoke the initial double-strand break to begin with. And there are many ways to deal with this problem. With zinc fingers, the approach is the placement on the repair construct of an additional selective mutation that is engineered to disable nuclease rebinding and recutting. But that issue aside, here are just two published examples which highlight with zinc fingers, but the same is true for all other nuclease platforms, how essential it is to cut at the mutation. We use zinc fingers because they're essentially designable against any position, and I'll show you a couple of examples this morning of how that's been useful clinically. This was a collaboration with Rudolf Janish's lab, and Dirk Hockemeyer actually contributed very productively to this, in which we repaired a mutation in alpha-synuclein by straddling the mutant site with engineered zinc fingers. And subsequently, we collaborated with Ludovic Vallier and Alan Bradley in targeted correction of alpha-1 antitrypsin. And here, again, the familiar theme, cut at the mutation or as close to it as you possibly can. So in approaching the mutation hotspot in IL-2R gamma, which causes bubble boy disease, we relied on the fact that the zinc finger is so flexible and built a zinc finger nuclease heterodimer that cuts essentially at the mutation hotspot. Because this work is published, and it's kind of surreal for me to think that it was published 10 years ago. Holy moly. I have vivid memories running these gels. I will walk you solely through the things that have stood the test of time. The first is that optimization of nuclease reproducibly makes editing better. Now, how you optimize is a function of what nuclease you choose. With zinc fingers, you tune the protein-DNA interface, so you tweak the alpha helices. This is one of the things that you can do. And what we showed back there is if you take two separate zinc fingers, an original one, which recognizes this stretch of DNA via this string of 4 alpha helices, and look at the efficiency of gene correction that it drives, and separately, you optimize the protein-DNA interface for affinity and specificity by making just 2-point mutations. You see quite a striking effect. And the effect is of a dual nature. First, the y-axis here is efficiency of gene correction. Optimizing the zinc finger dramatically improves the efficiency of correction, dramatically. Only 2 amino acid changes, you go from that signal to this signal. We also have an indirect but clear readout that this optimization improves the specificity as well, because the original zinc fingers, when put into the cell, drive correction, but the fraction of corrected cells drops by 50% after 7 days of culture. In contrast, the optimized zinc fingers, the frequency of corrected cells in the pool stays flat. So as you think about your favorite editing application, whatever your nuclease of choosing, it can always be better from an on-target potency and from a specificity perspective. And with zinc fingers, fortunately, there's a very clear path to attaining that improvement because the protein-DNA interface is tunable. All right, and so that's lesson number 1. Lesson number 2, double-digit correction at endogenous loci in human cells should be and is routine with no selection and in 24 hours. Now, back then in 2003, 2004, when Mike Holmes and I got this result, which you're about to see, it took us about 6 months to believe ourselves. We would make up devil's advocate arguments why this is an artifact and would do controls, because efficiency of this sort had no precedent in the literature at all. What do you mean 20% of the chromosomes in your cells at the endogenous locus have acquired this novel SNP without selection in 24 hours? Are you kidding? Well, we were not. But so as you think about your editing application today, what this took was not just an optimized nuclease that cuts precisely at the target, but also a repair construct and an expression construct for the nuclease that was delivered to the cells in an optimal way. So the teachable moment to all of you, experienced and aspiring editors, use a positive control. Whatever cell you work with, whatever critter, whether you work with the rat or the sea squirt, look at the literature. The editing literature dates 15 years. Find an example where your favorite critter or cell had been edited with high efficiency, double digit, no selection, single digit days, and deploy that protocol. The expression construct, if you're doing correction or integration, the modality of delivery of the repair construct, etc. Don't settle for anything other than really sky-high numbers. Second teachable moment, this was a PCR assay 4 days later. We saw up to 20% correction. The other teachable moment that we learned quite quickly, especially when we grew out these cells to 30 days and did this by Southern blotting, because again, you realize back then, this seemed quite futuristic. So we wanted to do this by Southern because we were deathly apprehensive about PCR-induced artifacts. Fortunately, the Southern blot showed that things are good. And the second teachable moment here is that, especially for modalities that involve HDR, cell cycle state matters, we discovered that arresting the cells in G2 dramatically increases the fraction of chromosomes that are resolved by HDR of the donor. I see in the literature today the rediscovery of this fact, which is reported as a novel finding, which is great. I'm delighted people are repeating that experiment. I'm very, very happy. But you should think about, this old adage that we all learn in graduate school. Don't use 2 months in the laboratory to save an hour in the library. So as you approach an editing project of your choosing, give some thought to the fact that the editing literature is actually quite extensive and quite old. Many different tricks and tools have been built and described. Look through it. It well may be the case that between zinc fingers, TALs, and Cas9, there's already a trick in the literature that gets you to the outcome of your choosing. But that said, it's always nice. It's always nice to repeat a finding that already exists. But if you can just copy paste it, Control-C, Control-V, that's faster. And last but not least, don't settle for heterozygous anything. You can homozygose the target for the mutation of your choosing. I don't know what adjective to use. We danced in the streets when we got the first human cells homozygous for a de novo introduced point mutation that we had specified, because that had clinical implications. This meant that we could correct not just recessive but dominant conditions as well. Single step. And what did it take? It took optimized delivery and cell husbandry. So you can and should, I encourage you, obtain cells or creatures, both heterozygous and homozygous, for the allele of your choosing. Because a comparison of this dosage effect, if you will, or of this series of organisms or cell lines could be biologically deeply informative. And I know those of you with a yeast or fruit fly or mouse background will say, well, you're preaching to the choir. My message to you is this should be the MO when doing somatic cell genetics as well. So there you have it. 2003, 2004, gene correction of the mutation hotspot in IL-2R gamma that causes bubble boy disease to 20% with no selection, with all these biallelic clones. Ta-da! Well, not really. Because what we then had to do, and I'm proud to report we did, is rise to the translational challenge of putting this into the clinic. How do you put a break into the clinic? And it took us 4 years from the publication in Nature. They asked us for a catchphrase for the cover. And we proposed the term genome editing because we thought that this technique really deserves to be described using a novel term. Nature accepted our suggestion, and I'm very pleased to say that, of course, that's the term that has been subsequently adopted by everyone, whatever nuclease you use, which is great. But Nature then said, well, we're going to rewrite the rules for gene therapy. And I remember seeing this going, right, now we just have to do that. Took about 4 years to file and get approval for our first investigational new drug application. Why did it take all that time? Well, because there's all these real-world issues that we faced on and took on, and I'll walk you through how we did some of that. There was if you will, the medicinal chemistry of the nuclease, the necessity to engineer potency and specificity into the nucleases that will be clinical grade. There was the need to develop, in collaboration with the FDA, and the RAC, which is the Recombinant DNA Advisory Committee of the NIH, an essential agency with respect to gene therapy, a regulatory path with respect to safety, both in vitro and in vivo, using multiple readouts. And last, but putting it very mildly not least, there was the issue of clinical deployment. How do you deliver? How do you manufacture it? And then ultimately, the clinical infrastructure to do a clinical trial. How do you actually put a break into a human being? So all of this happened. As I'm about to tell you, we've treated more than 70 subjects by genome editing, and I'll give you a set of unpublished observations from our clinical experience so far, and give you a preview of where we are with respect to the clinic. So I'll walk you through, as promised, HIV, the hemoglobinopathies, clotting factor diseases, and lysosomal storage disorders. So our initial foray into genome editing in the clinic, in fact, did not focus on gene correction, and I'm happy to explain why. It focused on HIV. So 2 decades ago, work here in the Bay Area described a cell surface molecule, CCR5, as being absolutely required for HIV to establish a productive infection of the human T cell. And of course, as you know, in rare individuals who are resistant, who are homozygous for loss of functional allele of CCR5, are resistant to infection by our tropic HIV. One person has been cured of his HIV infection. The HIV community does not like the word cure. They call it the C word because they're very apprehensive of making claims that are unsupported. One person has been cured, Timothy Brown, the famous Berlin patient, and he's been cured because he received a bone marrow transplant, fully ablative, so eradicating his own immune system and hematopoietic system, from an individual who's homozygous for delta 32, this loss of functional allele of CCR5. So this suggested for us immediately a plan for clinical action. Obviously, finding, there are 39 million HIV positive people on the planet. There are 1.2 million HIV positive people in the United States. Finding HLA matched donors for all of them who are homozygous for the delta 32 allele of CCR5 is not an option. But we thought, what we could do is we could obtain T cells from an HIV positive individual, use genome editing of the CCR5 gene to knock it out, and then return the cells to the patient. And I put the word simple in the subject in quotation marks because it's almost sweet what we didn't know when we set out to do this. Now, ultimately, we did this with some really remarkable findings. But my other sort of suggestion to you is as you venture on and use something, don't overthink the feasibility, because if you do, you might scare yourself into inaction. Sometimes it just makes sense to take the plunge. We did, and here we are. So the first step was engineering clinical grade activity and specificity into our nucleases. And of course, you're all familiar with the process, through which a small molecule such as Gleevec, shown here, emerges out of an iterative process in which you take an initial scaffold and then you tweak it by the addition or removal of various moieties, and you get this wonderful clinical drug. Now, so we rely on the zinc finger. I just realized that my pointer is pointing on the screen, but not on this screen. On tonight's episode of Unfriendly Laptop. So we use ZFNs, as I mentioned, because zinc fingers are extraordinarily engineerable. A ZFN, as you learned from Dana and everybody else in this workshop, is a heterodimer of 2 DNA binding domains. And we have a large amount of opportunity for optimization of this entity for on-target potency and specificity. We have multiple modules. We have multiple ways of linking them together and multiple ways of linking the zinc finger array to FOC1 to tune how this agent binds to the target locus and to teach it away from binding to other loci. So the essential toolbox, and again, I understand that most of you right now are not working with zinc fingers right now, but as you think forward to whatever nuclease platform you're planning to use in the next 30 or 50 years, what it took for zinc fingers to succeed clinically was a large and extensively characterized library of modules, a set of module linkers that allow us the targeting a given sequence with dozens of different module configurations, and linkers that allow us to skip different spacer lengths. And all of them are available to us as we approach a given clinical target. And even then, the path we've adopted is a multi-stage one, in which we begin by scanning a target for initial hits and assemble research stage molecules that we typically screen in transform cells, such as K562s. But then through multiple steps of iterative improvement, we tweak the nuclease such that they are maximally potent with respect to cleavage at the target and maximally specific with respect to genome-wide profile of action. And as we do this, as we tune for potency and specificity, we transition the screening to the more clinically relevant cell types. And I'll show you several examples of how we actually did this for the clinic, just to give you a sense of what it took to put a double-strand break into a cell that then went into a human being. Sir?
So I have before you move on to a specific sort of conceptual question, taking the med-chem analogy, very frequently in drug discovery, potency, specificity, or are orthogonal to what eventually becomes the drug. And there are all kinds of other parameters that go in. So the most potent, most specific compound is a great tool, but is not the drug for various reasons. Have you seen any evidence of things like that with these kinds of modules? The most specific, the most type binding set of modules is not the thing that you would eventually translate.
We have. So Jacob is asking, is it sometimes the case that the most potent and the most specific is not the one that for some reason we take into the clinic? The best example I can offer comes from our work on Huntington's disease. And I don't have the slides, so I'll do this briefly, but it actually touches on a really good point. For Huntington's, our goal is to discriminate between the wild type and mutant allele. And the way we've done that is by engineering modules that bind CAG, 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 CAG. And as you all know, that's what Huntington is caused by. And what we found is the most potent and specific CAG binders bind the wild type allele, which we want to protect, and the mutant allele equally well. So in fact, our most favorite zinc finger array is not the tightest and most specific, in part because we wish to go after the more expanded version rather than the contracted option version. With respect to more traditional nuclease targets where we don't need such exquisitely fine discrimination between two allelic forms of the same gene, I don't think we have enough experience in the clinic and advanced preclinic. Potency is our first goal. Typically, things that are the most potent in transformed cells port quite nicely to primary cells. It is sometimes the case that high potency comes at the cost of an excessive burden of off-target. But what we typically do is we take multiple early stage ultra-potent leads into specificity testing. And at that point, we basically select the most specific one while normal of comparable potency. So in other words, we don't have to have that trade-off. But Jacob is asking a great question. This is the experience of drug discovery has many a lesson in which sort of in vitro or simple cell model-based readouts for function can sometimes deceive you. So thank you. Any other questions or thoughts about this? Okay, and what do we have for time? 9:23. Coffee break coming up soon. Let's do this. Let me walk you through the HIV clinical data, and then we'll stand up, stretch our legs, have some coffee. All right. We want to knock out CCR5. And so Mike Holmes and Ed Rebar at Sangamo led the effort in building clinical-grade zinc finger nucleases that cut CCR5. And I'll talk at great length about them because they form a really robust study of what you can learn about editing in the clinic. And of course, when introduced into primary human T cells or hematopoietic stem cells, they induce a break at the target, and the signature of the break is, of course, these small insertions and deletions. And these are useful for two reasons. The first, of course, is that they interrupt the CCR5 open reading frame, creating loss of function mutations. And the second is they create a permanent signature of zinc finger or nuclease action, which you can track in the clinic. And we've done that. So step one, of course, was the development of how do you deliver the nuclease to primary human CD4 T cells. And after a very substantial amount of work, we settled on a viral delivery with hybrid serotype adenovirus at 535. And we've subsequently developed messenger RNA delivery of the nuclease as well, which we're testing in the clinic. The generally teachable moment for all of you is non-trivial cell types, such as primary human T cells or hematopoietic stem cells, may require more than your mini-prep DNA nucleaffected into your cells by an AMAXA shuttle. So if your editing efficiency or viability is below what you desire, delivery alone could make a substantial difference. And it absolutely does for many, many cell types. Well, fine, we've knocked out CCR5. Now what? Well, so a theme that we'll run through this morning is the use of in vitro and animal models of efficacy. And they're an essential step on the path of any therapeutic or preclinical concept to more advanced work. Here, it was essential to show that this de novo CCR5 knockout, which you realize when we did this, it had never been done before. A de novo editing, any kind of de novo knockout in primary human cells of anything had ever been done before. And to say nothing about the fact that these are primary human T cells and a gene, the knockout of which supposedly should endow the cells de novo resistant to entry by the virus. And this is the slide of death. There's just an unpalatably large amount of information. The bottom line, you take some mice which have been configured to allow the transplantation of human T cells, and you either edit the cells or not at CCR5, and you stick them into mice, and then you infect or not the mice with HIV, and you ask for two things. What happens to the virus load, and what happens to the T cells? So the base state, which you see here, is that in the absence of HIV infection, the input frequency of editing, whatever was in the cells that you put in, should basically stay flat. And it does. Because in the absence of the virus, the edited cells have no survival advantage until you infect with the virus. And what the virus does is it kills the wild types and selects for the edited ones. So this was good to see. But what happens to the virus? So fine, so these resistant cells grow out in the mice. So what happens to the animals is the viral load drops. Why? Because the virus requires a productive infection. It doesn't really live very well outside the cell. It requires a productive infection to continue to multiply. And as the immune system is repopulated by a compartment that's resistant to de novo infection, virus titer drops. So this was good news for us. And so what we then did is, greatly encouraged by these and a wealth of other findings, we developed a GMP compliant, and because this is a workshop more focused on basic science applications, I will not take you through the years and years of work, literally, that it took to develop a GMP compliant process. So in order to do this in the clinic, you have to do this under good manufacturing practice. The basic idea was that you take a patient with HIV, you harvest T cells by apheresis, you expand them, you induce a disruption of CCR5, you expand them further, test them rigorously, and then you infuse them back, and their clinical dose on the trial has been between 10 and 30 billion. So, in extensive work and an extensive discussion with the agency, I'm proud to report that we have charted a regulatory path for non-clinical safety that the agency has reviewed and accepted. As I mentioned to you, we've treated over 70 subjects with their own edited T cells, and I'm also very proud, very genuinely proud to report that we have 2 open INDs that the agency reviewed and approved for targeted editing in hematopoietic stem and progenitor cells. So the reason this is relevant is this means that agency review and understanding of editing has grown substantially, and that there is precedent with respect to the expectation that the agency will set for you if you approach it with a desire to cut somebody's chromosome. So if I go through the next 10 to 20 slides slowly, it will take me an hour. So in the interests of time and sanity, let me just give you a high-level survey of what are the kinds of things that we've done in order to go to the clinic and to get INDs approved. To determine the overall functional impact on the cell with respect to the safety of the break, we have a wealth of assays that sort of phenotype the cell. We fax them, we'll look at growth kinetics, look at cytokine release, diversity of the TCR. These will be specific to your specific cell product. But for T cells, if you're interested, here are some data from a published paper that you can look up to see how did we scale up the assessment of safety on the T cell. Then transitioning to molecular assays of, and I know Dana talked about off-targets, and in fact the notion of off-targets has been an important one to look at since the very early days of editing. You know, the obvious hope is that editing allows the promise of site-specific modification, and of course conventional gene therapy by definition, every single cell that is clinically relevant has an off-target effect because every single cell has a random integrant. But there are unequivocally potential risks to the genome. There is undesired DSB generation at sites beyond the intended target, and of course genomic rearrangements driven by the induced DSB. So there is an integrated set of assays that one uses to look at the fidelity of editing. An assay that we've done is enumerate double-strand breaks, which can be done by looking at the chromatin marks in control cells, cells treated with our nucleases, or cells treated with a control agent. This work is published, so I'll simply summarize that when you take zinc fingers to disrupt CCR5, you reproducibly see one double-strand break above background. So, but this is a very sort of pan-nuclear assay. What about more genome-directed approaches? Not surprisingly, these have evolved over time. An initial assay we developed that has been quite useful and robust is to determine the actual in vitro preference of DNA binding by the zinc fingers, and you can do the same for TALs. How to do this for Cas9 is an interesting question. We can certainly discuss this. And then you identify the actual the actual preferred consensus binding site for the zinc finger, and in this way of displaying the data, this is the sequence that the left and the right zinc finger were designed against, and this is a collation of in vitro binding studies in which all the signal above the bar is where the zinc finger recognizes the intended base, and all the signal below the bar is where it recognizes, in addition to that, something else. So you can see the overall biochemical specificity of these agents is quite robust. Why is this useful? So now, okay, fine, you have this. What do you do now? Now you scan the genome, not just with a mere match to intended, but with a match to what the zinc fingers actually bind. And I know Dana spoke about Keith Jung's work on GuideSeq. I don't work on Cas9 daily, but my reading of Keith's paper is that the GuideSeq data conclusively demonstrate that Staph aureus Cas9 with a single guide cleaves in the nucleus in a manner that cannot be explained by bioinformatics. And I don't know if that's an overstatement, but at least that's the conclusion that I see in Keith's paper. So something for all of you to ponder. So the way we've approached this is by using this biochemistry biased assay in which we identify the PWM for the individual zinc fingers. Then we scan the genome for a match to what the zinc fingers actually bind. And then we use deep sequencing to genotype them, those sites in actual cells treated with a nuclease. And when we've done this, we identify in this specific experiment two off-target sites. One is a paralogous gene CCR2, and one is an off-target site called ABLIM1, which is a non-coding one. This has unequivocally a looking for the keys under the lamppost modality to it, which is why we developed, based on an extensive precedent in this regard in the gene therapy field, an unbiased or a less biased approach, which is based on end capture. And our initial work on this was published back in 2011. And of course, as I just mentioned, the Guide-seq paper was recently published, which follows very closely on the fundamental notion of this. We've elaborated on this assay and developed it, and I'll tell you about some of our more recent findings as the talk progresses. The long story short is you treat the cells with a nuclease and an agent, a nucleic acid that can be captured in the break, and then you scan your cells for where the entity landed, and you ask, well, where did it go? And we reproducibly try to do this at maximal doses of the nuclease where the cells are still alive. If you will, we wish to create the worst case scenario, so maximum tolerable on-target marking. And this is published, so I'll just summarize everything you see on this slide in one sentence, which is we see low single-digit numbers of additional off-target sites, but most remarkably, and this was really exciting to us and important to us, and again, because this is published, I'll go through this quite quickly. When we look at the off-target sites that these nucleases cut, the first and most important point is none of them appear to be coding or, based on all of our analysis, associated with a known bioinformatic risk. When we look at the DNA sequence of the loci that are cut by the zinc fingers, they're all explicable in terms of what we know about the biochemistry of the zinc finger. So zinc finger biochemistry, as gauged in vitro, appears to drive the specificity of pan-nuclear action. So, which is really comforting. That means that they continue to behave as the designed designed DNA binding proteins that they are. In the interest of time, I'll skip through the next 3 slides, simply to say that there are 2 standard important assays that allow you to look at whether treatment of cells with your zinc fingers creates something untoward. There's a clonogenic assay, which is established, and for our clinical leads, it was negative. And then there is a soft agar transformation assay, which is again established. To give you a sense of how potent these things are, and this was done, I would imagine, in 2006, 2007. In these fibroblasts, the zinc fingers drove 80% targeted knockout, targeted disruption, I should say, at the highest multiplicity of infection, and no anchorage-independent growth was observed. So last, but very significantly, in collaboration with the agency, we developed an in vivo assay of safety. And in single sentence terms, the assay involves manufacturing a clinical scale dose of your intended cell product using your clinical lead nuclease, then engrafting the cells into immunodeficient mice in a way that maximizes the potential for these cells to become transformed and to develop a tumor, and then allowing the mice to live as long as is biologically relevant with respect to obtaining a readout of carcinogenicity. So the logic is, if there's something the zinc fingers or any other nuclease is doing that is carcinogenic, well, let's park them in mice for as long as we can and see if any tumors develop. And as you can imagine, we would not have taken this into the clinic if this study that we did with our clinical cell product did not come back clean. Namely, that when this study was done, I realize there's a very substantial amount of text here. The bottom line is, I'll skip again in the interest of time to the single most important slide. When we did this study, no CCR5 modified CD4 T cell derived neoplasm was seen. So I told you that in de-risking this approach on the path to the clinic, we did both standard in vivo safety assays as well as novel assays we developed, which is site selection to predict the top sites and monitor them by deep sequencing, as well as an unbiased and capture-based assay. We did tumorigenicity of a full patient dose of cells in immunodeficient mice. And together, these data provided both the molecular and phenotyping data supporting the safety of ZFN-modified CD4 positive T cells. And so in 2009, we were able to initiate the first trial. And since I suspect most of you don't work on HIV or on adoptive T cell therapy, I'll just give you some very high-level perspective on what we found. And then for the rest of the time available to me, I'll show you what other kinds of things we're doing as we're thinking about advancing this to the clinic. By the way, I'm speaking really fast, but this is just because I'd love to tell you more. By all means, if you have a question, just please raise your hand, or don't even raise your hand, just say something. I would love to answer any questions any of you may have. Sir.
I don't know if you're going to address this, Fyodor, but
yeah.
when you're looking at CCR5, and I know you guys basically tile down the gene looking for ideal sites.
Yep.
When you pick that site, so where is it? You know, is the first half close to the very start of the first exon? What's the most common in HEJ you see? Is it insertion, deletion,
Yep.
1, 2, 3? And then how variable is that? How how wide of a repair difference do you see?
Yeah, it's a great. Thank you for. So I'll repeat the three questions for the tape. So where did we cut in CCR5? How do you position the target site for the nuclease? Second, with respect to repair outcome, is there a common allele? Third, how broadly diverse is the allelic spectrum that's going into the patient? As a matter of practice, we try to go for the first 1/3 of the ORF. In our case, I mentioned to you that there is a naturally occurring allele. I don't know if I have that schematic. There's a naturally occurring allele of CCR5 called delta 32, which creates this truncated form, which is non-functional. So we knew that that was a safe place to go. We went upstream of that. As some of you may know, CCR5 is a single, the coding region is a single exon. One of the really useful features of many human genes is there is nonsense-mediated decay. So a double-strand break placed in essentially, and a premature stop that occurs in any exon other than the penultimate, upstream of the penultimate one will lead to the message to be degraded. The caveat there, of course, is that some messages in the human genome are NMD resistant. We were delighted to see that a common outcome of the repair at CCR5 is the duplication of the 5 base pair overhang, that the 5 nucleotide overhang that the nucleases leave in vivo. So a very common event that accounts for up to 30% of the alleles in the cell product is in fact a duplication of that site. So there's a 5 base pair insertion, which of course is a knockout. And that's been useful to us with respect to tracking the cells clinically. And dozens, if not hundreds of alleles are present in the cell product. So the outcomes are very, very diverse. Sir?
Curious about the cohorts.
Yep.
When you got these cohorts, did you guys for specific KIR genes, HLA genes, mutations in KIR genes?
So the question is, was any screening with respect to any immunological genotype done?
KIR3DS1, for instance, is associated with a lower myeloma.
Yes. So the answer is, did we screen for KIR32DS with respect to potential for lower viral load? The initial cohorts were enrolled purely based on immunological responder versus immunological no responder, and in a subsequent trial, as I'm about to explain, whether or not the patient is or is not heterozygous for delta 32. But strategically, if I may sort of walk down that tangent for a bit, you know, we all know about pharmacogenomics, right? Vemurafenib, which was developed by Plexicon just down the road here, which is this wonder drug against melanoma, is developed against a specific allele of BRAF, I guess V600E or something like this, right? Even for drugs that are not developed in a targeted fashion against specific alleles, you know, there's clear evidence that the genotype of the individual affects the outcome of the therapy. I mean, I think a prime example is CYP2D6 polymorphisms and tamoxifen and the response of a subject on the trial, of a woman to tamoxifen therapy. I think the pharmacogenomics of response to genome editing therapy is a wonderful topic to dig into. We have data on 70 subjects so far, so that, to the best of my knowledge, in fact, it's safe to say, those are the only people who have been genome edited in clinical trials in the Western world. I fully expect, as time goes on, as these therapeutics expand, that inclusion and exclusion criteria and enrollment parameters will be tweaked precisely in the spirit of the question that you're raising, both with respect to likelihood of response to the therapeutic at the disease biology level. So for example, is there a pre-existing polymorphism that controls viral load, something like this, as well as potentially responding to the editing act itself. We just need more data, but this is a great question. Stay tuned to this space. Please.
Is there any thought about targeting the integrated HIV directly?
Yeah, people have done this. The question is there any thought to targeting the proviral genome? Absolutely. And there's some nice work discussing this in the literature. I think this has potential. As you doubtless know, the overwhelming majority of your T cells live in your gut. So gut-associated lymphoid tissue, or GALT, forms the bulk. I mean, we think of our blood as being where it happens. In fact, our immune system lives in our gut. So as far as eradicating the virus, you would need a way to get to some cells that, the robust eradication of the target would be an interesting challenge to face on. We chose to go after, actually this is a good point because it sort of highlights why we went after CCR5. The hope was to create a compartment of the immune system that's protected against de novo infection. That was the plan. And so both our T cell trial and our CD34 trial are focused on that. But I don't want to disparage the notion of pursuing the integrated provirus by editing, simply to raise the fact that you will have the challenge of getting to the cells. What's been the clinical experience so far? I'll give you some high-level summaries. In our initial effort, and this is published, I'll go through this very quickly and then talk about unpublished work. We enrolled 2 cohorts. These are all folks with HIV. They're all on antiretrovirals, and some of them have been on antiretrovirals for quite some time. And one thing you should understand is that, there are wonderful drugs that control viral load and control active disease. But when you talk to actual folks who have spent 5 or 10 years taking cocktails for highly active antiretrovirals, if they could stop taking them, they would. If they could stop taking them, they would. So we focused on 2 cohorts, folks whose CD4 count is above 450, so-called immune responders, and folks who after HIV infection and then the control of active viral replication by antiretrovirals, whose T cell count did not rebound, and who are thus so-called immunologic non-responders. So we developed, as I mentioned to you, a GMP-compliant manufacturing process. We would take CD4s from an individual, expand, edit, expand further, freeze, release for infusion, and then transplant. The published work, briefly. And so you'll see a lot of bar graphs. There's a lot of graphs that look like this. And the Cartesian coordinates typically will be time on the x-axis and something interesting on the y-axis. So the first key finding was that in both cohorts, T cells bearing a disruption of CCR5 persisted in the circulation for out 250 days. As you're about to see, our longest marked subjects are now out to 4 years and beyond. So it is kind of remarkable. There are people walking the planet with nuclease-induced indels in their immune system for the past 4 years. And so far, the treatment in all the subjects has been well tolerated to date. Well, fine, so we have marked cells. And so what? The second key finding, and this is really significant, is that in a majority of the subjects, infusion of their own disrupted CCR5 loss of function bearing alleles led to a sustained and significant increase in CD4 T cell count. I'm about to show you the primary numbers in a second, but let me just emphasize in the strongest possible terms that for a subject who is an immunologic non-responder and is below 450, an increase of 150 to 200 CD4 cells per microliter above baseline is clinically quite significant. Now, a key hypothesis of our effort was that if CCR5 is in fact essential for HIV infection of T cells in vivo, then a de novo of disruption of this gene should lead to their protection, right? And so to ask whether these T cells in fact create an HIV compartment of the immune system, I'll talk about more of this in a second, we performed what's called an STI, or a structured treatment interruption, where we actually took away their medication. And this was part of the clinical protocol. The subject consented to this, and this was done under very rigorous safety standards with respect to human subject protection. And we noticed immediately that during that treatment interruption, as viral titer rises, wild type and modified cells were eradicated by the virus at a much more rapid rate than cells bearing a modification of CCR5. So this was really encouraging. What I'd like to do now, and this and other data are in the New England Journal paper, what I'd like to do is show you some unpublished work that creates a more comprehensive picture of our clinical findings to date. We recruited a cohort of immunologic non-responders. So these are people with whose viral count is in the blood below detection, but their T cell count is below 450. And that is a problem. That is a problem. And so this was a dose escalation study with respect to how many cells we infuse. And the two significant findings that I'm thrilled to report to you this morning is this adoptive transfer of T cells following disruption of CCR5. Now, this is one of those graphs that is very common in clinical trial data presentation, but basic scientists, such as myself, find very hard to look at because it's unclear how to parse this. Each line is a person. The x-axis is time all the way up to month 44. The y-axis is CD4 count above baseline. And you'll just have to take my word for the fact that we have a durable increase in CD4 count in the majority of subjects at 3 years plus post-infusion. And the mean increase, in fact, is clinically quite significant. 162 CD4 T cells per microliter, which is a mean increase of 45% from baseline. And for an immunologic non-responder, clinically speaking, that's huge, just from a quality of life perspective. The second finding is that CCR5 modified T cells persist 3 years post-single infusion. And the x-axis has stayed the same, and the y-axis is what's happened to the marked cells. Perhaps most strikingly, and I'm showing you data on 9 subjects, you should understand that these are people with a substantial HIV reservoir. They are on heart, so they have no actively replicating virus in their bloodstream. But their virus reservoir is very, very substantial and is a lifetime one. And normally just stays flat. And we were absolutely delighted to observe in 6 of the 9 subjects on the trial a 0.3 to 3.6 log decrease in the HIV reservoir. So this means that through mechanisms that I can speak about at some length if you wish, this adoptive transfer of CCR5-bearing knockout T cells leads to a drop in the overall body-wide load, if you will, of HIV reservoir in these individuals. So the clinical findings so far, we've reconstituted the CD4 compartment and sustained 3 years plus post-infusion. CCR5 modified cells detected 3+ years, and we have a substantial decay of the HIV reservoir. I saved the best for last, and it is that in a number of the subjects of our trial, we have observed that the subjects are able to control their viral replication in the absence of continued antiretroviral therapy. So the single-time infusion of CCR5 disrupted T cells allowed them to control their virus without taking their medication, other than the modified T cells that are now circulating in their bloodstream. And to walk you through how this is done, I need you to show you the timeline. So on day 0, we infuse the genome-edited T cells. Then day 28, we stop antiretrovirals as per protocol, and this is fully consented, of course. And then we monitor over the next few weeks, we monitor viral load in the bloodstream. So I'm going to click a button, and you're going to see a whole bunch of curves, and each of which is viral load in an individual over time. And so the thing you see, which is really astonishing to me when I first saw these data, because I'm not an HIV person by training, is how rapidly the virus rebounds. Some of these people have been HIV positive for 10+ years. Before you stop ART, virus is below detection in their bloodstream. You stop the meds, and within a week or 2, virus rebounds to 10 to the 4 to 10 to the 5th. It's been waiting here for 10 years for you to stop the medication. Thank you very much. And we were, I don't know what word to use. I'm actually at a loss. It's hard to explain what that feels like. I'll just say it. Several of the subjects on the trial experienced a reduction in viral load before resumption of antiretrovirals. And one of them, this person you see here, the viral load dropped to below detection of the assay before this person had to resume antiretrovirals as per protocol. So this was the dream. This was the plan to have this happen. What made this person special? We didn't know it at the time, now we do. This person is a heterozygote naturally for that loss of function allele of CCR5. And you're all experienced or aspiring editors and understand why this is happening. The nucleases had only one allele of CCR5 to get rid of, to create a fully knockout cell. So, this is an experimental therapeutic. And in advancing it forward, we have the opportunity to structure clinical development in a way that is guided by our previous experience. So we were able to enroll and treat a cohort of individuals who are heterozygous for the delta 32 allele of CCR5 and are HIV positive. And they are actually quite common. And so I'll show you the results of our studies of one such individual who is identified here but just by this number. But the bottom line is that this person's baseline viral load before the meds set in was north of 10 to the 3. This person received an infusion, I actually don't know the gender, I'm sorry, of 10 to 30 billion CCR5 modified T cells. Then this person stopped taking their antiretroviral medication. Virus titer initially rose, then flattened out, and has stayed flat at 255 particles per mil, which is considered non-infectious, out to the last reportable point, 900 days. So this person is controlling their virus without antiretroviral medication. We realized that engraftment of biallelically marked cells, so cells in which both copies of CCR5 are knocked out, thus might be associated with a more favorable clinical outcome. And to attain that, we had the luxury of an existing drug, Cytoxan, which can be used to precondition the subjects and create room in their peripheral circulation with respect to the transplanted T cells. So we basically make space in their hematopoiesis for the T cells that we infuse. I'll skip through all of this clinical detail in the interest of time and simply show you one piece of data. And this one piece of data are a number of subjects. Each bar is a subject on the trial. And the axis is the log of the viral load drop from peak to the end of the treatment interruption. And with an exception, the subjects experienced a drop in the viral load before resuming antiretrovirals. But in a number of the subjects, specifically 1, 2, 3, 4, they remain free of their HIV meds because their viral load is either below detection or below a level that is considered infectious. So I've shown you that we've reduced to clinical practice, and you realize this is an ongoing clinical effort. I'm just giving you some highlights. That reduces to practice this flowchart that you saw earlier, in which you start with folks with HIV. The target is CCR5. You use zinc finger nucleases that you've optimized to clinical grade activity and specificity to induce a double-strand break at the target, which resolves by disruption. And I've shown you durable effects in the clinic on marking CD4 count and anti-HIV effect. So we are continuing the clinical development of this because HIV is not a simple disease. It is not a simple disease. Creating an HIV-protected compartment of the immune system is not a trivial challenge. But the fact that we have durable effects on CD4 count, clear effects in a number of the subjects on viral load despite the subjects remaining on ART, and perhaps most strikingly, a number of subjects whose viral load is either now below detection or who are stably controlling their viral replication, despite no longer taking antiretrovirals, is tremendous grounds for optimism. So before I dive into the next bit, happy to answer any questions that I can. This is not a simple topic. So I won't be able to give you comprehensive answers to everything that may be lurking in your minds, but I'd love, if anybody has any questions, happy to pose and answer them. Sir?
So if you were to irradiate like the patient who was cured, would you expect to see a sort of similar cure in some sense? Like if you irradiated all the natural T cells before you started this?
So the question is, can you do, are you proposing to do an autologous bone marrow transplant? So harvest bone marrow to protect this person from dying from lack of hematopoiesis? then park the cells ex vivo while you irradiate and then put them back in? I'm embarrassed to admit, I don't know what the clinical record of that specific approach has been. My understanding is that the sort of the side effects of a total body irradiation and a fully ablative conditioning in an adoptive autologous transplant setting are so severe that it is routinely, I'm not a clinician, so to be clear, is anyone in the room a physician? Are you?
But yeah, there is, significant side effects. We have heart, which we'd much rather give the patient than an autologous liver transplant. Also, I think there's reservoirs of mature T cells that are not annihilated by the ablative therapy. So in this case, you might not achieve that.
So to recap what the doctor in the audience said, is there a doctor in the house? Thank you, doctor. So what we heard was there are reservoirs that are not eradicatable using standard chemotherapy approaches. And as I hypothesized, the adverse event and the sort of the morbidity mortality profile associated with a fully ablative conditioning and a total body irradiation is just simply the safety profile is not compatible with the fact that these folks are actually antiretrovirals that control the virus.
I think there's clinical trials that cover where they did something similar to the Timothy Brown patient, the bone marrow transplant, but without the CCR5.
Yep.
And then those patients relapsed after 1 or 2 years.
That's right. So the other thing, thank you, you're absolutely right. So this lady pointed out that clinical trials, small scale, have been done in which folks have received fully matched transplants of just healthy marrow rather than delta 32, well, I'm sorry, I take that, I don't mean healthy, wild type marrow, not delta 32 homozygous knockout. And unfortunately, those patients relapsed. So the virus came back. Other questions? Sir?
This is an expensive therapy.
Yes.
And each patient has to be treated individually.
Yes.
What are you thinking about making this more transportable, more affordable, so that it can be distributed more broadly?
Dana is asking a question that has haunted my dreams and thoughts for the past 15 years, and that's always a preamble for the fact that I don't have a good answer. But here's my best bet, my best stab at this. The clinical infrastructure required for this is very substantial. And furthermore, it has to be done GMP. And the moment you say GMP, you add however many zeros you can imagine to the price of whatever it is you're doing. So I cannot say anything about the cost of this treatment if and when it is approved, because that's not my neck of the woods. I can tell you that the one gene therapy approved right now in Europe is $1 million. I can tell you that the one adoptive T-cell therapy that is approved for prostate cancer, which is Provenge by Dendreon, is $100,000. These costs, how to scale these kinds of costs, and of course this kind of infrastructure, to not just the 1 million or so folks in the States, but the tens of millions of folks elsewhere where there are no GMP clean rooms, I think is for me, if you ask me, what is the challenge? That is the challenge. Now, lots of really interesting things are happening with respect to, for example, delivery. Are we constrained forever to processing T cells ex vivo in sterile bags? That's one thought. My sense, the way I've thought about this, early steps versus subsequent steps. We all remember the initial iPod, which was this sort of Walkman-sized device which weighed about a pound or something. Now, the iPhone, the miniaturization and technology advancements that have, this is essentially a highly glorified iPod plus a phone, right? And what did that take? It took iterative technological tweaks once the initial proof of concept has been established. So our current focus is very much on demonstrating clinical efficacy of editing. And we are sort of encouraged by the fact that pharma has adopted en masse the notion of biologics as a therapeutic. Great interest exists in pharma with respect to, for example, cancer immunotherapy. So this is cell-based therapies. So we are hopeful that as we advance the effort through the clinic, we get a good there will be lots of supporting effort elsewhere with respect to other kinds of things. And I just didn't answer your question, right? But you should be aware of the fact that this is something that's very much on our minds. No, that's a great question.
I was glad to hear that you're looking for your fingers before you put them in the clinic for off-target effects by this ideally captured technology. And they have covered many now other technologies for detection of off-targets that are being used or that have been applied to CRISPR-Cas9. But that there is a lack of kind of unifying study, kind of find which one is really kind of the best one and to detect the most potential off-targets or not. Is there, do you expect that there's going to be a standard for, as these gene therapies are coming for doing such comparison analysis, and that there's going to be a requirement for all gene therapies to do that off-target analysis?
So Jacqueline is asking a great question. 10 years from now, when genome editing is available from your local gas station, there won't be any gas 10 years from now, right? I mean, we'll all be on solar. Will there be a firm, robust standard that is applied uniformly across the board to the clinical deployment of genome editing for ex vivo and in vivo therapy? Absolutely. And I say this with such conviction because we're basically following a path that has been followed by other experimental therapeutics over the past 20 or 30 years, like a biologic. If you have a biologic you wish to put into the clinic for a given type of indication, the non-clinical safety efficacy path that you have to put through is a charted one. So what will it look like for nucleases? Well, you realize there's, the clinical experience right now is limited to HIV for T cells. We have 2 open INDs. One, I'm about to describe both of them. And the overall outline of the regulatory requirements with respect to safety are pretty clear, and are some of the ones that I described to you right now. What additional or less, whether the agency will demand additional effort or less than what we've done so far, I think will be a function of the clinical track record of the therapy. I think that's fairly safe to say. The agency and the clinicians will monitor the safety and efficacy of the editing trials that are ongoing, and that will continue to inform what the agency asks for. You asked me, will there be a standard? I certainly flatter all of us at Sangamo into thinking that in some sense, at least right now, for ZFNs, for T-cell and CD34 therapy, I don't want to say standard, we've established a precedent that the agency has reviewed and approved in both open trials and open INDs. But I would be misrepresenting the situation if I looked you and they said, and I promise you that this shall be the standard that shall persist for the next decade. So for now, we have a one step at a time, walk before we run approach. Yeah, absolutely. So the lady's asking, are we planning to redose? Absolutely. And one of the advantages of, I mentioned that we can deliver in the form of messenger RNA. One of the advantages of redosing, of messenger RNA delivery rather than virally based delivery, is there is absolutely unequivocally the potential to redose the subjects. Absolutely. Okay, so it looks like I have, with a Carol allotment of additional 5 minutes, I have maybe 14 minutes or so, and I'm halfway done. Let me give you sort of high-level summaries from the hemoglobinopathies, the hemophilias, the lysosomal storage diseases, and finish on my favorite quote of all time. We spent an enormous amount of time trying to deploy editing in the context of T cells. We have subsequently worked out how to drive very efficient knockout in human hematopoietic stem and progenitor cells. And somebody over coffee break, and I'm sorry, I forgot your name, asked me about why not edit CD34 cells, because we can protect other compartments of the hematopoiesis, such as dendritic cells and macrophages, from HIV infection. Absolutely. But what that required is being able to deliver nucleases to CD34s in a way where it can manufacture a clinical dose and get high efficiency knockout. And I'm delighted to report we did that. I will skip through the efficacy data because they're published and tell you what it looks like. It looks like electroporation using in vitro transcribed capped polyadenylated GMP-grade messenger RNA. And the electroporator is something called a Maxite GT, and the reason we use it is it is clinical grade. It has a device master file on file with the FDA. And I'm very pleased to report that we have 2 reviewed, approved, open INDs for use of this device with mRNA electroporated in human CD34s. In our program to disrupt CCR5, we get north of 40% CCR5 disruption achieved in mobilized HSPCs at clinical scale production. So the teachable moment for all of you, whatever your nuclease of choosing, is if CD34s are your cup of tea, mRNA, and electroporation. And so I will spend 3 or 4 minutes giving you a very high-level summary of how we have begun to approach preclinical and then clinical development for the hemoglobinopathies, so sickle cell disease and beta thalassemia. We're very fortunate to be partnered with Biogen in this program. So everything I'm about to describe is in close partnership with them, and it's thrilling to be partnered with big biotech because, of course, of the experience they bring to the table. To the best of my knowledge, we will become the first organization in the history of biomedicine to translate findings from a genome-wide association study directly to clinical practice. So symptoms of both sickle and thal, which of course are caused by mutations in beta globin, can be suppressed or even eliminated if that individual has an additional mutation which elevates her or his levels of fetal globin, which live elsewhere in the genome, but in most of us are silent. In some people, it's as high as 20%. And if they have that, and if they have a mutation that causes sickle or thal, their disease will be much milder or will be eliminated. So this was discovered in a genome-wide association study, and what was discovered is the master regulator of this protection against disease is variation in a gene called BCL11A, which is a transcription factor which acts to repress fetal globin. So we've translated this to a reviewed open IND by developing clinical-grade zinc fingers that bind to and disrupt BCL11A. This again required a highly sophisticated ex vivo cell processing sequence. And an important slide I'd like to share with you is, if you build them, they will come. If you engineer the zinc fingers just right and optimize delivery, then you can reproducibly get round numbers 70%, 70, single-step targeted indels in a clinical scale dose of human HSPCs. 70. This is what tuning of the nuclease and optimizing delivery can do. So don't settle for low single digit. Editing is a powerful thing. I will skip through, in the interest of time, all the preclinical evidence for efficacy and safety that led ultimately to us having an approved and open IND. In very broad terms, we followed the charted path drafted for us by the CCR5 experience. So the safety package was very similar and included both in vitro and in vivo assessments of safety. And of course, we had an efficacy package to contribute as well. I want to spend 2 minutes telling you about a recent development which really highlights the marvelous new world of being able to use genome editing against any druggable target. I mentioned to you that the world of genome editing now means that we can drug anything. So the actual genetic variation that underlies resistance to sickle cell disease or beta thalassemia symptoms doesn't lie in the coding region of BCL11A. It lies in an erythroid-specific enhancer of this gene, as discovered by Stu Orkin's lab. Now, this was found quite a while after we'd already advanced our clinical program for the targeted knockout of BCL11A. Then lo and behold, this paper comes out, and they say, well, if we can knock out the enhancer, then this could get all the benefits of elevating fetal globin while sparing the effect of losing BCL in other lineages, which would have an effect on B cells. We thought, well, we're fast. Why don't we just build clinical reagents to knock out the enhancer? So go from a state in which we knock out BCL11A everywhere to just editing an enhancer, so a regulatory element, and thus confine the effects of the knockout to just the erythroid lineage. I'm very happy to say the work has been submitted for, accepted for publication, so it's coming out soon. So in the interest of time and wishing to spend at least 5 minutes on in vivo editing, I'll skip through all of the data to give you a short punchline. This complex regulatory element, which consists of 3 separate stretches of DNA, which are bound by who knows how many transcription factors, appears to contain an Achilles heel. And that Achilles heel is the target site for an erythroid-specific transcription factor called GATA1. And by tuning to the base pair, the position at which the ZFNs induce the double-strand break, and placing the position of the break as close as we could to the center of this Achilles heel. And by optimizing the zinc fingers further, I am delighted to report that we've been able to attain a functional effect, so an elevation in the level of fetal globin from knocking out the enhancer that is equivalent essentially to what we see by knocking out the gene itself. So let me pause for 30 seconds and say this again to say how remarkable this is. You knock out a gene, just the coding region, you get a dramatic functional outcome, elevation of fetal globin. Surprise. Or you knock out one position in a lineage-specific regulatory element of that gene, and you get the equivalent functional readout. That, to the best of my knowledge, has no precedent in the history of the literature. That has never been accomplished before. And I am delighted to report that in partnership with Biogen, that is the approach that we're planning to take to the clinic for beta thalassemia and for sickle cell disease. I told you about the use of ex vivo delivery of nucleases to T cells in the clinic and to hematopoietic stem and progenitor cells for the treatment of sickle cell disease and beta thalassemia. What I've not had an opportunity to talk to you about, some of this is published, some of this I'm hopeful is coming out, is the fact that we've optimized nucleases and their delivery to allow the efficient editing of the liver. And in partnership with Shire, we're advancing that as a treatment for the hemophilias. The idea is we switch editing modalities. It's no longer targeted knockout, it's targeted integration. And the idea is that you deliver to the body virus, which routes itself to the liver. One virus delivers the nuclease, the other virus delivers the corrective payload. Upon cleavage, the corrective payload integrates, and we've developed a safe harbor, a safe harbor locus in the human genome, albumin actually, which allows us to use it, if you will, as a USB port of the human genome, in which we can plug and play any ORF, and that plug and play process yields robust levels of protein secreted into the bloodstream. So we're advancing that to the clinic for the hemophilias, and separately for the lysosomal storage diseases. Now, biologically, they're very different. But what is common among them is they're all currently treated, to the best of anyone's ability, by enzyme replacement therapy. So this intravenous infusion or other routes of administration of recombinant protein, which is expensive and challenging. The strategic plan, this is sort of the next step in genome editing, is to have albumin be a platform for in vivo protein replacement. So if you can treat something by enzyme replacement therapy, you can deliver the editing machinery systemically such that it routes to the liver, integrates the desired cassette into the albumin locus, and the liver thus becomes the factory, if you will, that produces the protein that then corrects the disease defect. And what I had to skip through in the interest of time is evidence of preclinical efficacy and safety with respect to being able to actually do this clinically. Last 2 slides. One of my idols, Sidney Brenner, said, progress in science depends on new technologies, new ideas, and new discoveries in that order, which I find remarkable. Brenner, of course, was the father of many brilliant ideas, so the fact that he prioritizes technologies over everything else is quite striking. We're the beneficiaries of living in an era of remarkable technology development. We have remarkable ability to sequence to understand the basis of disease, and we can now manipulate the genome. The genome is remarkably tractable. So that's thrilling for all of us. So last quote, and then I'll just show you an acknowledgement slide. You know, the popular press is it's slightly amusing to read it. they've very rapidly identified the hazards of editing as the issue du jour. The current issue of Atlantic Monthly lists genome editing as one of the technologies that we might regret having invented. I mean, it literally says that. I go, interesting. And I'm not questioning that, as all technologies, this has the potential of causing something that we won't be happy with. But I'm surprised that what's been overlooked in all of this is what remarkable are the implications for the management of genetic, infectious, and malignant disease are. So my angle on this is the last slide before I show the acknowledgements, is a wonderful quote from Condorcet, who is one of the founders of positivism. And he goes, and if you read French, I encourage you to read it in French because it's better in French than in the translation, because that, to me, describes the future of genome editing in the clinic for the next 10 to 20 years. I've undertaken work that shows through reasoning and facts that nature has assigned no limit to the perfecting of human faculties, and that the progress of this perfectibility has no limit other than the duration of the globe on which nature has placed us. Now, doubtless its progress will be more or less rapid, but never will humans retrograde, or so as long at least as the Earth occupies the same place in the system of the universe. Yeah. And most important slide, just a few high-level thanks. Carl June led the early-stage clinical trials for HIV. The HSPC work was done with Paula Cannon. We have the enormous honor of working with George Stamatianopoulos and his son John Stamatianopoulos at UW on beta thal. I'm very grateful to Haiyan Zhang and Kasra Kasrin from Biogen for their help. Folks at Shire, Kathy Hai for their work at Children's Pennsylvania on hemophilia. And of course, I wouldn't be here without 15 years worth of shoulder-to-shoulder effort at Sangamo. Abduribar, Jeff Miller, Li Zhang design and build the zinc fingers. Andreas Reich leads the hemoglobinopathy effort. Gary Lee, the HIV work. My comrade in arms, Michael Holmes, is remarkable. It's been a thrill working with him for all this time. Jinbin Wang and his group does some work I had to skip over on efficient targeted correction in HSPCs. Thomas Wexler and Russell DeKelver work on the lysosomal storage disease and hemophilias. Philip Gregory, good colleague, good friend, has resigned from Sangamo recently, but I must acknowledge the tremendous contribution he's made. And Dale Lando and Jeff Nickel are clinicians who lead this all into the clinic. Thank you for letting me go over time. Thank you for the invitation. Thank you for the wonderful questions.